Hi, everyone. Today I'll be talking about how hormones can influence the behavior of honeybees both by organizational and activational effects. First of all, we're just going to go over what JH is. It stands for juvenile hormone, and it's secreted by the corpora allata endocrine glands located behind the brain in honeybees. And we've seen in class how JH levels actually control transitions between key stages of the honeybee life cycle. For instance, it's involved in the timing of nurse to forager transition in worker bees. So as we discussed in class, these forager bees will return to the hive and expose the young bees to ethyl oleate. And this exposure causes them to not produce JH and continue that nursing activity within the hive. However, as these older foraging bees die off, there's less ethyl oleate exposure, which leads to higher JH production within those younger bees and activates that foraging behavior. So in class, we really focused on this example as a way to express how social interactions can lead to behavioral changes, but it's also a great example of how hormones can have both organizational and activational effects on behavior of honeybees. So just to go over the difference, organization really focuses on how hormones can reorganize the brain to make permanent changes, but this must occur within a specific critical period of that animal or insect's stages of life. However, hormones can also have an activational effect by triggering a behavior at any stage in the life cycle. So JH can have organizational effects in honeybees by altering gene expression to cause both polymorphisms and polygenisms within honeybees, and this can lead to intraspecific variation in morphology and behavior. And a great example of this is actually through caste determination between the queen bee and the worker bees. So the queen bee is first picked out as the female larva with a high metabolic rate. And so once that's distinguished, during the third larval stadium, that queen larva is fed a sugar and protein rich diet that initiates that JH production. And then during their fifth larval instar feeding stage, we see a high JH peak. And that's actually the critical period for that JH to act on and distinguish that queen bee. And this early spike in JH actually results in a smaller mushroom body in the queen compared to workers. And that's important because mushroom bodies play a role in learning, which is important for worker bees as they leave the hive. We also see activational effects of JH in honeybees because it determines the division of labor among workers by triggering that nursing to foraging behavior. So as we discussed earlier, that lack of ethyl oleate allows for JH release, which then activates young bees to begin that foraging behavior. We also see that JH can influence age-related responsiveness to the alert pheromone. And so that's just another great example of how JH hormone release later in honeybees' life stages is an example of activating response to alert signals. So we see that in forager bees, they have increased gene expression for enzymes responsible for the conversion of methyl farnesylate into JH. And so as they develop, there's an activation from JH of these genes, specifically looking at the MFE gene. This is activated by the foraging carbohydrate-rich diet that's different from the nursing bees' protein-rich diet. And that carbohydrate-rich intake helps lead to JH production. So the MFE gene is a great example of this age-related switch to foraging and the differences in nutritional physiology of foragers compared to nursing bees. We also see MFE's role displayed in the transgenic honeybees that had MFE knockdown as they produced less JH in honeybees. So in conclusion, hormones can have both organizational and activational effects in response to nutritional intake during development, as we saw in queen bees versus worker bees, as well as in response to chemical triggers released by social interactions, as we saw from the nurse to foraging transition. Thank you so much.